Hi, I'm Rochelle Crosby Watson, Professor of Integrative Biology and Physiology, and we're going to continue on our journey in skeletal muscle physiology and look at the actin and myosin interactions that constitute the Crossbridge cycle and that lead to skeletal muscle force. So, where we last left off was our discovery of the sarcomere. And remember that the sarcomere is composed of these Z lines that define the boundaries of the sarcomere. And within each sarcomere, you'll notice that there are thin filaments and thick filaments. We discovered those during our last mini lecture. And we know that those thick and thin filaments overlap when skeletal muscle contracts. And that observation led to a second hypothesis at the time. And that hypothesis was that muscle contraction results from changes in the thick and the thin filaments and the interactions between those filaments that shorten and produce force simultaneously. So, scientists then set out to do an experiment in which they measured the force produced at various sarcomere lengths. The experimental setup is shown schematically for you here, and we also have this great demonstration of the experiment where we can measure the force produced by a skeletal muscle at various sarcomere lengths. The sarcomere length is determined by stretching the skeletal muscle out at the outset of the experiment before the skeletal muscle is stimulated to contract. So we hope that the frog video really helps you understand this experimental setup and how we're able to very specifically measure force produced by skeletal muscle. So, when scientists did these experiments and measured the force produced, shown on the y-axis, as a function of sarcomere length depicted on the x-axis, I hope that you can see that maximum force-generating capacity, i.e. the top of that curve, is generated when there's this maximum overlap between the thick filaments, specifically those regions where there are these protrusions, and the thin filaments. So this is called a force or tension length curve, and it's very commonly used in skeletal muscle physiology. So, the conclusions from those experiments was that there was a relationship between the sarcomere length and the force-generating capacity, such that the shortening of skeletal muscle occurred by an overlap between those thick and thin filaments, and that it was the overlap that was generating the force that is needed to contract in skeletal muscle. And I'm going to show you now a few illustrations, and you can play with these at home. So here is a relaxed sarcomere. Look at the structure of the Z lines. Find those for yourself. Look at the thick filament. Now in this illustration, the thick filaments are drawn with these protrusions from them. Those are called the myosin heads, or the head structures. We're going to look at those in detail in a moment. And notice how those protrusions have the ability to interact with the thin filament. And in this particular diagram, they're showing that one thick filament is surrounded by 6 thin filaments on each side of those head structures, each side of those protrusions. So this is the relaxed sarcomere. Take a look at the contracted sarcomere. So what's happening? Those Z lines are moving closer together. There's no change in the overall length of the thick and the thin filaments, but there is certainly an increase in the overlap of the thick and the thin filaments. So, how does that happen? Scientists have spent decades investigating the molecular mechanisms of skeletal muscle contraction, and it first started by asking the basic question, well, gee whiz, what is actually in the thin filament? What are the components, the protein components that make up the thin filament? So biochemists took skeletal muscle and they purified the thin filaments by a series of centrifugations and dissociations. And at the end, as you can see on the bottom of your screen, what they discovered is that for every 350 proteins that they called actin, there were 50 proteins called tropomyosin and 50 troponin proteins. And that structure, again, was very regular and very repeated. That stoichiometry of 7 actin molecules to 1 tropomyosin to 1 troponin complex was consistent in skeletal muscle. And it's these other proteins, the tropomyosin and the troponin, that are regulating the interaction of actin with myosin to produce skeletal muscle contraction. Now, while we have this illustration up, I just want to point out that the individual balls that you're seeing that polymerize together to form the actin structure are really like 2 strands of pearls that are twisted upon each other. I also want to point out while we're here that tropomyosin is a very elongated protein, and it binds along the length of the actin filament. Troponin is actually a complex of 3 proteins called troponin T, troponin I, and troponin C. And those bind at regularly spaced intervals to tropomyosin into actin. Okay, so how is the thin filament formed? Well, the thin filament is formed from individual G-actin or monomeric actin, also known as globular actin, when these individual monomers come together, polymerize to form this gorgeous elongated filament structure. That filamentous structure is called F-actin or filamentous actin. And then it's on this filamentous actin that tropomyosin and troponin are built. And it's the interaction between these thin filament proteins that allows for the regulation of skeletal muscle contraction. So scientists took the same approach to investigating the molecular composition of the thick filaments. They took skeletal muscle, dissociated it, purified out the thick filaments, went through a series of centrifugations and dissociations to purify proteins, and they identified what they called as myosin heavy chain, which is simply a polypeptide protein, and myosin light chains. And they discovered a really unique ratio that every myosin light chain was interacting with a myosin heavy chain, but in fact, there were 2 myosin light chains that interacted with the myosin heavy chain. So in the schematic diagram shown here, there are 2 myosin heavy chains that are wrapped around each other. And the region that's being wrapped around, this coiled coil region, is the tail of the myosin heavy chain. And then there is a globular structure which represents the head of the myosin. You can see that there is also a regulatory light chain and an essential light chain that are interacting around the neck region of the myosin. We won't discuss the roles of the essential light chain and the regulatory light chain, but to suffice it to say that they allow for an extra layer of regulation. So, what they also discovered, and this is an entirely fascinating area of science, we discovered that molecular motors are actually a part of biology. And what that means is that a molecular motor is a protein that can take the energy from, for instance, ATP and convert it to mechanical movement. This is really spectacular. There are many examples of molecular motors. Myosin is one of them. So the myosin head is able to bind to ATP, hydrolyze it to ADP and inorganic phosphate, and then take the energy from that hydrolysis and change the structure of myosin to allow it to have these large dynamic motions. Okay, so it's myosin that takes the energy, ATP, converts it to structural conformational changes, and that allows the myosin head to reach up, interact with actin, which is part of the thin filament, and pull the Z lines closer to the M line. So that structure is shown here. So we've looked at the structure of the thin filament, which is shown in the upper portion of the diagram. We've looked at the structure of myosin, which is shown on the lower portion of the diagram. And it's the interaction between the two, or the physical contact between the two, that drives skeletal muscle contraction. And that's shown for you here. So the actomyosin contact is what we often refer to when we talk about the Crossbridge cycle. And the Crossbridge cycle is this wonderful series of complex molecular events that translates into this wonderful movement of the myosin head as it's contacting the thin filament and pulling the thin filament toward the M line. Okay. So what we're going to watch now is this great instructional video that shows you the actin and myosin interactions that constitute the Crossbridge cycle. Watch the video, and then we're going to break down the various components of the cycle.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the Crossbridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Now that you've watched a video, here's a review question. Shown on this schematic diagram are 4 individual stages of the cross-bridge cycle. Label each stage and describe how they contribute to skeletal muscle force production. Think about it. The answer is shown here. For the first stage, the myosin head is in a cocked back position. In this position, the myosin head has already hydrolyzed ADP and inorganic phosphate, and that conformational change is evident in a ratcheting of the myosin head backward. In step 2, the myosin head reaches out and grabs onto the actin binding site on the thin filament, on actin itself. Then the actomyosin interaction is changed as the head ratchets forward in what's called number 3, the power stroke. Finally, ADP comes and binds to the myosin head, and it causes the myosin head to release from the thin filament. And again, the myosin head will hydrolyze ATP again, and the whole cycle repeats itself, just as you saw in the previous video. And each of these myosin heads is binding to actin, hydrolyzing ATP, pulling forward in the power stroke over and over again, as long as there are signals that tell the actin and the myosin to continue on in the cross-bridge cycle. So, we just learned the basic elements of the actin and myosin interactions that constitute the cross-bridge cycle. Scientists have spent decades dissecting the individual molecular events, the conformational changes in all the proteins that we've discussed. And there's so many conformational changes that lead to the power stroke, that lead to force production. Scientists have even used little microatomic tweezers to investigate the individual force produced by single myosin heads. So this really is a fascinating area of research, and I hope that you've enjoyed it. And we will see you in the next mini lecture to talk about how calcium regulates the entire cross-bridge cycle.